The animal kingdom never ceases to amaze us with its boundless diversity. From the soaring heights of eagles to the depths of the ocean where bioluminescent creatures dwell, life on Earth presents a breathtaking tapestry of forms and functions. But what happens when these distinct branches of the evolutionary tree intertwine? The possibility of different species interbreeding, blurring the lines of classification, has long captivated our imaginations. Could a lion, with its majestic mane, truly sire an offspring with a tiger, the striped phantom of the jungle? Is the mythical centaur, half human and half horse, merely a product of fantasy? Or could there be a kernel of biological truth behind the legend? The concept of hybridization, the interbreeding of different species, takes us beyond the familiar boundaries of the natural world and into a realm where the very definition of a species is challenged. It compels us to confront fundamental questions about evolution, genetics, and the very essence of life itself. Can creatures as seemingly disparate as a horse and a donkey produce viable offspring? And if so, what does this tell us about the intricate mechanisms that govern life's blueprint? Our journey into the world of hybridization begins with a deceptively simple question. What exactly constitutes a species? In the intricate web of life, where subtle variations can be observed even within a single population, drawing clear lines between species is no easy feat. The traditional definition, rooted in the concept of reproductive isolation, posits that a species is a group of organisms capable of interbreeding and producing fertile offspring. However, as we delve deeper, we'll discover that nature often defies such rigid categorizations. Join us as we explore the captivating world of hybridization, unraveling the genetic mysteries behind these unusual unions and examining the evolutionary forces that both drive and constrain the intermingling of species. From the well-known mule, a testament to the endurance of hybrid vigor, to the more exotic ligers and tigons, we'll uncover the secrets of these biological marvels and delve into the ethical considerations surrounding human intervention in the delicate balance of nature. The quest to define a species, a seemingly straightforward task, has puzzled naturalists and scientists for centuries. The sheer diversity of life, with its countless forms and adaptations, makes drawing clear boundaries a complex endeavor. One of the most widely accepted definitions, known as the biological species concept, hinges on the principle of reproductive isolation. This concept, championed by the renowned evolutionary biologist Ernst Mayr, posits that a species is a group of populations whose members can interbreed in nature and produce viable, fertile offspring. This definition elegantly captures a fundamental aspect of species: their ability to maintain a distinct genetic identity over generations. If individuals from different populations can freely interbreed and produce fertile offspring, their genes continue to flow within the larger gene pool, ensuring a degree of genetic cohesion. Conversely, if two populations are reproductively isolated, meaning they cannot or do not interbreed, their gene pools remain separate, leading to divergence over time and potentially the formation of new species. However, the simplicity of the biological species concept belies the complexities inherent in the natural world. Nature, it seems, often delights in defying our neat categorizations. The concept falters when confronted with organisms that reproduce asexually, such as bacteria, or when considering extinct species known only through fossils, where reproductive behaviors remain a mystery. Furthermore, hybridization, the interbreeding of distinct species, throws a wrench into the neat definition, blurring the lines we attempt to draw between species. Despite its limitations, the biological species concept provides a valuable framework for understanding the concept of species and the forces that drive speciation. It highlights the crucial role of reproductive isolation in maintaining the integrity of species and serves as a springboard for exploring the myriad ways in which life has diversified over millions of years. Yet, as we delve deeper into the intricate tapestry of life, we must remain cognizant of the exceptions, the grey areas where the boundaries between species become blurred, reminding us that nature often defies simple categorization. While the biological species concept provides a useful framework, the reality of species boundaries is far more fluid than a rigid definition might suggest. Nature rarely adheres to our neat classifications, and the lines between species often blur, particularly when considering the vast timescale of evolution. Hybridization, the interbreeding of distinct species, stands as a prime example of this fluidity, challenging our understanding of species as discrete entities. Consider the case of ring species, a fascinating phenomenon that illustrates the gradual nature of speciation. Ring species are distributed in a circular pattern around a geographic barrier, with adjacent populations capable of interbreeding. However, as one moves around the ring, genetic divergence increases, and at the point where the circle closes, the two populations at the ends are often so distinct that they cannot interbreed, effectively behaving as separate species. Furthermore, the process of speciation itself is often a continuum rather than a series of discrete steps. Populations may diverge genetically while still maintaining some degree of gene flow through occasional interbreeding. This fuzzy boundary between species is particularly common in plants, where hybridization is more frequent than in animals. The existence of cryptic species further complicates matters. Cryptic species are morphologically indistinguishable, meaning they look nearly identical, yet are genetically distinct and reproductively isolated. These hidden species, only identifiable through molecular analysis, highlight the limitations of relying solely on physical characteristics to define species boundaries. The fluid nature of species boundaries underscores the dynamic interplay between evolution, genetics, and the environment. Species are not static entities, but rather points along a continuum of genetic divergence and reproductive isolation. Recognizing this fluidity is crucial for understanding the complexities of biodiversity and the processes that shape the tapestry of life on Earth. The animal kingdom, in all its splendor, rarely fails to surprise us. While we typically categorize creatures into distinct species, nature often colors outside the lines, producing remarkable exceptions that challenge our understanding of biological boundaries. These exceptions, known as hybrids, are the result of interspecies breeding, a phenomenon that piques our curiosity and offers a glimpse into the intricate mechanisms of genetics and evolution. Hybrid animals, bearing traits of both parent species, embody the fusion of different evolutionary paths, often resulting in unique appearances, behaviors, and capabilities. One of the most captivating aspects of hybrids is their unpredictability. The blending of genetic material from different species can lead to a remarkable array of outcomes, some more successful than others. In some instances, hybrids exhibit what is known as hybrid vigor, surpassing their parent species in size, strength, or resilience. The mule, a classic example of hybrid vigor, demonstrates exceptional strength and endurance, making it a valuable work animal for centuries. However, the story of hybrids is not always one of triumph. Hybridization can also lead to reduced fertility or even complete sterility, as seen in the case of mules, which are typically unable to reproduce. This reproductive barrier underscores the genetic complexities of interspecies breeding and highlights the delicate balance of genes required for successful reproduction. The existence of hybrid animals, both successful and challenged, raises intriguing questions about the nature of species, the limits of genetic compatibility, and the evolutionary forces that both drive and constrain the emergence of new forms of life. As we delve into the world of real-world hybrids, we'll uncover the scientific underpinnings of their existence, exploring the genetic mechanisms that govern their traits and the evolutionary implications of their presence in the intricate web of life. The animal kingdom holds a particular fascination for the offspring of lions and tigers, majestic creatures whose roars echo through our collective imagination. These hybrids, born from the union of a lion father and a tiger mother, are known as ligers, while the offspring of a tiger father and a lion mother are called tigons. These imposing creatures, with their striking blend of stripes and potential for massive size, capture our attention and spark curiosity about the interplay of genetics and the boundaries of species. Ligers, often exceeding both parents in size, stand as testament to the phenomenon of hybrid vigor. These massive felines, with males sometimes reaching over 1,000 pounds, inherit a growth-promoting gene from their lion fathers and lack a corresponding growth-inhibiting gene from their tiger mothers, resulting in their impressive stature. Their coats often exhibit a mesmerizing blend of their parents' patterns, with faint stripes adorning a tawny background, occasionally punctuated by a mane inherited from their lion lineage. Tigons, on the other hand, tend to be smaller than their parent species, often exhibiting a more slender build than their liger counterparts. Their coats typically feature a more prominent display of stripes inherited from their tiger fathers, with the possibility of fainter spots or rosettes reminiscent of their lion mothers. While Tigons may not achieve the colossal size of ligers, they nonetheless embody the captivating allure of hybrid animals, showcasing a unique combination of feline grace and power. The existence of ligers and tigons, while captivating, highlights the complexities of interspecies breeding. These hybrids, primarily found in captivity due to the limited overlap of lion and tiger habitats in the wild, often face health challenges related to their unusual genetics. Furthermore, their reproductive capabilities are often compromised, with males typically sterile and females exhibiting reduced fertility. These limitations underscore the delicate balance of genes required for successful reproduction and the evolutionary barriers that often prevent widespread hybridization in the wild. The humble mule, a familiar sight in rural landscapes and historical narratives, stands as a testament to the enduring partnership between humans and hybrid animals. Born from the union of a male donkey and a female horse, the mule embodies the concept of hybrid vigor, exhibiting exceptional strength, endurance, and sure-footedness that have made it an invaluable work companion for centuries. Its existence speaks to the practical applications of hybridization and the ways in which humans have harnessed the unique traits of hybrid animals for our benefit. The mule's remarkable strength stems from its inheritance of the donkey's robust physique and the horse's powerful musculature. This combination, coupled with their typically larger size compared to donkeys, allows them to carry heavy loads and navigate challenging terrain with relative ease. Their endurance, a testament to their hybrid heritage, enables them to work long hours, often under harsh conditions, without succumbing to fatigue. Furthermore, their sure-footedness, a trait inherited from their donkey fathers, makes them particularly well-suited for navigating mountainous or slippery terrain. Despite their many advantages, mules are typically sterile, a consequence of the genetic mismatch between their horse and donkey parents. Horses possess 64 chromosomes, while donkeys have 62. This discrepancy results in mules inheriting an odd number of chromosomes, 63, which disrupts the normal pairing process during meiosis, the cell division process that produces gametes, sperm, and eggs. This chromosomal incompatibility renders most mules incapable of producing viable offspring. The mule's sterility, while a biological limitation, has not diminished its value to humans. Their strength, endurance, and sure-footedness have made them indispensable work animals in various cultures and industries, from agriculture and transportation to mining and exploration. Their legacy serves as a reminder of the profound impact hybrid animals can have on human societies and the enduring fascination with the potential of hybridization. The realm of hybrid animals extends beyond the familiar liggers and mules, encompassing a captivating array of creatures that blur the lines between seemingly disparate species. Zebroids, the offspring of zebras and other equine species, and holphins, the rare hybrids of whales and dolphins, exemplify the remarkable diversity of hybrid animals and the intriguing possibilities that arise when different branches of the evolutionary tree intersect. These unusual pairings, often occurring in controlled environments or as a result of chance encounters in the wild, offer a glimpse into the fluidity of species boundaries and the unexpected outcomes of genetic mixing. Zebroids, with their striking striped patterns inherited from their zebra parentage, showcase a remarkable variety of coat variations depending on the other equine parent involved. Zorses, born from the union of a zebra stallion and a horse mare, often exhibit a blend of stripes and solid colors, while zonkis, offspring of a zebra stallion and a donkey mare, may display a more muted stripe pattern against a lighter background. These captivating creatures, prized for their unique appearance and potential for disease resistance, often inherit a combination of their parent species traits, such as the zebra's hardiness and the horse's or donkey's temperament. Holphins, on the other hand, represent a rare and fascinating example of hybridization in the marine world. These hybrids, typically resulting from the union of a false killer whale, a member of the oceanic dolphin family, and a bottlenose dolphin, exhibit a blend of their parents' physical characteristics, often possessing a body size and shape intermediate to their parent species. Their coloration may also reflect their hybrid heritage, with a blending of the darker tones of the false killer whale and the lighter greys of the bottlenose dolphin. While holphins are not commonly found in the wild, their existence underscores the potential for hybridization even among seemingly disparate marine mammal species. The captivating world of hybrid animals, with its ligers, tigons, mules, and other fascinating creatures, presents a compelling paradox. While these hybrids showcase the potential for interspecies breeding and the blending of distinct lineages, they often face a significant hurdle: sterility. This reproductive barrier, a common theme in the world of hybridization, highlights the delicate balance of genetics required for successful reproduction and the evolutionary forces that often limit the long-term success of hybrid lineages. The root of hybrid sterility often lies in the mismatch of chromosomes between parent species. Chromosomes, the thread-like structures that carry genetic information, must pair up correctly during meiosis, the cell division process that produces gametes like sperm and eggs. When parent species have different numbers of chromosomes or significant structural differences in their chromosomes, their hybrid offspring inherit a mismatch that disrupts this pairing process, often rendering the hybrids infertile. This chromosomal incompatibility acts as a genetic barrier, preventing the widespread establishment of hybrid populations. While hybrid animals may thrive as individuals, their inability to consistently produce viable offspring often limits their evolutionary potential. This reproductive isolation, enforced by genetic mechanisms, helps maintain the integrity of distinct species and prevents the blurring of genetic lines over generations. However, the story of hybrid sterility is not always one of absolute barriers. In rare instances, fertile hybrids may arise, often through complex genetic mechanisms that overcome the initial chromosomal incompatibility. These fertile hybrids, capable of producing offspring, have the potential to bridge the genetic gap between their parent species, introducing new gene combinations and potentially contributing to the evolutionary trajectory of both lineages. At the heart of life's intricate tapestry lies the chromosome, a microscopic, thread-like structure that carries the genetic blueprint of an organism. These remarkable structures, residing within the nucleus of every cell, orchestrate the symphony of life. dictating everything from an organism's physical traits to its susceptibility to certain diseases. Each species possesses a specific number of chromosomes arranged in distinct pairs, with one chromosome from each pair inherited from each parent. Humans, for instance, possess 23 pairs of chromosomes, totaling 46, while chimpanzees, our closest living relatives, have 24 pairs, totaling 48. This seemingly subtle difference in chromosome count between humans and chimpanzees, a mere 2 chromosomes, represents a significant genetic barrier, underscoring the importance of chromosomal compatibility for successful reproduction. When 2 individuals from the same species reproduce, their chromosomes align and recombine during meiosis, the cell division process that produces gametes like sperm and eggs. This intricate dance of genes ensures that the resulting offspring inherit a complete set of chromosomes, maintaining the genetic integrity of their species. However, when individuals from different species attempt to interbreed, the choreography of chromosomes becomes far more complex, often leading to missteps and misalignments that can disrupt the delicate balance of genetic information. The resulting hybrid offspring, inheriting a mix of chromosomes from their parents, may face challenges ranging from reduced fertility to developmental abnormalities, depending on the extent of the genetic mismatch. This chromosomal dance, while often depicted as a straightforward pairing process, is in reality a highly orchestrated event involving a complex interplay of molecular actors and cellular machinery. The success of this intricate process hinges on the compatibility of the chromosomes involved, highlighting the crucial role of chromosomal harmony in maintaining the boundaries between species and the challenges faced by hybrid organisms that inherit a genetic mosaic from their parents. While the number of chromosomes plays a crucial role in determining the compatibility of 2 species for interbreeding, it is not the sole arbiter in the intricate world of hybridization. Beyond the sheer quantity of chromosomes lies a complex landscape of gene regulation, a delicate interplay of molecular switches and dials that fine-tune the activity of genes, determining when, where, and to what extent they are expressed. This intricate regulatory network ensures that genes are activated and silenced at the right time and in the right cells, orchestrating the development and function of an organism with remarkable precision. Imagine a symphony orchestra, with each musician representing a gene, poised to play its part in the grand musical composition of life. The conductor, in this analogy, represents the intricate system of gene regulation, ensuring that each instrument enters at the precise moment, playing at the correct volume and tempo to create a harmonious melody. Just as a misplaced note or an ill-timed crescendo can disrupt the flow of music, so too can disruptions in gene regulation lead to developmental abnormalities or even disease. In the context of hybridization, differences in gene regulation between parent species can pose significant challenges, even when chromosome numbers align. The hybrid offspring, inheriting a mosaic of regulatory elements from its parents, may experience a clash of genetic instructions, leading to disruptions in gene expression patterns. These disruptions can manifest in a variety of ways, from subtle alterations in protein levels to more profound developmental abnormalities, depending on the genes affected and the extent of the regulatory mismatch. Furthermore, the intricate interplay between genes, with some genes influencing the activity of others, adds another layer of complexity to the hybrid equation. A gene that is normally tightly regulated in one parent species may be expressed at a higher level in the hybrid offspring due to the influence of regulatory elements inherited from the other parent species. This altered gene expression pattern, while not necessarily detrimental in itself, can disrupt the delicate balance of cellular processes, potentially leading to unforeseen consequences. Section 3. When species collide. Exploring genetic incompatibilities. Hybridization, the union of different species, often results in offspring that face an uphill battle for survival and reproduction. This struggle is rooted in the intricate dance of genes, where even subtle differences in the genetic makeup of parent species can have profound consequences for their hybrid progeny. These genetic incompatibilities, often invisible to the naked eye, can manifest in a variety of ways, from reduced fertility to developmental abnormalities, highlighting the challenges faced by organisms that inherit a genetic mosaic from their parents. One common source of genetic incompatibility stems from differences in the structure and function of proteins, the workhorses of the cell. Proteins encoded by genes carry out a vast array of tasks, from catalyzing biochemical reactions to transporting molecules across cell membranes. When parent species have evolved different versions of a particular protein, their hybrid offspring may inherit a mix of these variants, some of which may not function optimally together. Imagine a team of construction workers attempting to build a house using a mix of tools and materials from different manufacturers, each with slightly different specifications. While some components may be interchangeable, others may not fit together seamlessly, leading to structural instability or inefficiencies in the building process. Similarly, in hybrid organisms, mismatched protein variants can disrupt cellular processes, leading to reduced fitness or even inviability. Another layer of genetic incompatibility arises from differences in the way genes are regulated. As discussed earlier, gene regulation ensures that genes are expressed at the right time and in the right cells. When parent species have evolved different regulatory mechanisms, their hybrid offspring may inherit a clash of genetic instructions, leading to disruptions in gene expression patterns. These disruptions can manifest in a variety of ways, from subtle alterations in protein levels to more profound developmental abnormalities, depending on the genes affected and the extent of the regulatory mismatch. The consequences of genetic incompatibility can be far-reaching, impacting the hybrid's ability to survive, grow, and reproduce. In some cases, hybrid inviability may occur, with the hybrid offspring failing to develop properly or dying prematurely. In other instances, hybrids may survive but exhibit reduced fertility, limiting their ability to contribute to future generations. The story of hybridization, already a complex tapestry of genetic interactions, takes on an even greater level of intricacy when we consider the emerging field of epigenetics. Epigenetics, literally meaning above genetics, refers to changes in gene expression that occur without alterations to the underlying DNA sequence. These changes, often influenced by environmental factors or inherited from one generation to the next, act like molecular switches, turning genes on or off or fine-tuning their activity levels without affecting the actual code of the genes themselves. Imagine a musical score representing an organism's DNA sequence containing all the notes necessary to play a symphony of life. Epigenetics, in this analogy, acts like the musician's interpretation, adding dynamics, phrasing, and emotional nuances to the performance without altering the notes themselves. Just as different musicians can interpret the same score in unique ways, so too can epigenetic modifications influence gene expression patterns, leading to variations in traits and characteristics. In the context of hybridization, epigenetic differences between parent species can add another layer of complexity to the already intricate process of genetic mixing. Hybrid offspring, inheriting a combination of epigenetic marks from their parents, may experience unexpected gene expression patterns, leading to novel traits or even disruptions in normal development. These epigenetic influences can manifest in a variety of ways, from alterations in coat color or body size to more subtle changes in metabolism or behavior. Furthermore, the environment itself can play a role in shaping the epigenetic landscape of hybrid organisms. Environmental factors such as diet, stress, or exposure to toxins can trigger epigenetic modifications that influence gene expression patterns, potentially leading to variations in hybrid traits depending on their surroundings. This interplay between genetics, epigenetics, and the environment highlights the dynamic nature of hybrid organisms and the challenges in predicting their characteristics based solely on their DNA sequence. The biological world, for all its diversity and splendor, is governed by a set of rules that, while sometimes bent, are rarely broken entirely. One such rule, crucial for maintaining the integrity of species, dictates that reproduction should occur primarily within rather than between species. To enforce this rule, nature has erected a series of barriers, both physical and behavioral, that hinder interspecies mating and the production of hybrid offspring. These barriers, known as prezygotic barriers, act as gatekeepers, preventing the fusion of gametes, sperm and eggs from different species and preserving the genetic distinctiveness of lineages. Imagine a castle representing a species with its inhabitants seeking to maintain their lineage and protect their genetic heritage. Prezygotic barriers, in this analogy, act like the castle's defenses, preventing outsiders from breaching the walls and intermingling with the inhabitants. These defenses can take many forms, from moats and drawbridges to vigilant guards and strategic fortifications, each designed to thwart potential intruders. One of the most straightforward prezygotic barriers is habitat isolation, where species occupy different ecological niches or geographical regions, reducing the likelihood of encountering potential mates from other species. For instance, a species of bird that dwells in dense forests is unlikely to cross paths with a species that prefers open grasslands, minimizing the chances of interspecies mating. Even if their paths do cross, behavioral isolation can come into play, with species-specific courtship rituals, songs, or pheromone signals acting as recognition mechanisms, ensuring that mating attempts occur primarily between members of the same species. In cases where habitat and behavioral barriers are breached, mechanical isolation can provide an additional layer of protection. The reproductive anatomy of different species often evolves in concert, ensuring compatibility between males and females of the same species. However, this intricate fit can become a hindrance when interspecies mating is attempted, with mismatched genitalia acting like ill-fitting keys in a lock, preventing successful copulation. Finally, even if interspecies mating does occur, gametic isolation can serve as a last line of defense. The gametes of different species, even if they manage to meet, may be incompatible due to differences in their surface proteins or other molecular recognition factors. This incompatibility prevents the sperm from fertilizing the egg, thwarting the formation of a hybrid zygote. In the intricate dance of life, where the boundaries between species are sometimes blurred, the successful union of gametes from different species represents a rare but not unheard of event. However, even when a hybrid zygote, the single cell that results from the fusion of sperm and egg, is formed, the journey towards becoming a viable, fertile offspring is fraught with challenges. These challenges, collectively known as postzygotic barriers, represent the second line of defense against hybridization, ensuring that even if a hybrid offspring is conceived, its chances of survival and reproductive success are often limited. Imagine a hybrid organism as a delicate seedling struggling to take root in the fertile ground of life. Postzygotic barriers, in this analogy, act like harsh environmental conditions such as drought, disease, or competition from other plants that threaten the seedling's survival and ability to thrive. These challenges, while not always insurmountable, often stack the odds against the hybrid organism, limiting its chances of flourishing and contributing to future generations. One of the most formidable postzygotic barriers is hybrid inviability, where the hybrid zygote fails to develop properly or the resulting embryo dies prematurely. This early demise often stems from fundamental genetic incompatibilities between the parent species, such as differences in the timing or coordination of developmental processes. The intricate symphony of gene expression that orchestrates the development of an organism from a single cell to a complex multicellular being is easily disrupted by genetic mismatches, often leading to fatal errors in the blueprint of life. In cases where hybrid offspring do survive to birth, they often face the challenge of hybrid sterility, rendering them incapable of producing viable offspring of their own. This reproductive barrier typically arises from mismatches in chromosome number or structure between the parent species, as discussed earlier in the context of mules. These chromosomal discrepancies disrupt the delicate process of meiosis, the cell division process that produces gametes, sperm and eggs, leading to inviable gametes or offspring with an incorrect number of chromosomes. Even if hybrid offspring manage to overcome the hurdles of inviability and sterility, they may still face the challenge of hybrid breakdown, where subsequent generations of hybrids exhibit reduced fitness or fertility. This decline in vigor often stems from the accumulation of deleterious recessive alleles, harmful versions of genes that are masked when paired with a dominant allele, but can manifest in homozygous individuals, those inheriting two copies of the recessive allele. The world of hybridization offers a captivating glimpse into the intricate mechanisms of genetics, evolution, and the very definition of a species. We've journeyed from the familiar mule, a testament to hybrid vigor, to the awe-inspiring liger, pushing the boundaries of size and strength. We've explored the genetic underpinnings of hybrid sterility, uncovering the delicate chromosomal dance that can either harmonize or clash when species intersect. Yet our exploration has only scratched the surface of this complex and often controversial subject. The question of whether humans, with our unique evolutionary trajectory, could ever interbreed with our closest living relatives, chimpanzees or bonobos, lingers in the background, shrouded in both scientific curiosity and ethical considerations. Could such a union, defying the boundaries that separate us, ever produce viable offspring? What would the existence of such a hybrid, a being caught between two worlds, reveal about our place in the grand tapestry of life? In our next installment, we delve into the depths of this controversial topic, exploring the scientific plausibility of human-primate hybrids, the ethical debates surrounding such endeavors, and the myths and realities that have fueled our fascination with this taboo subject for decades. Join us as we venture beyond the hybrid horizon, confronting the profound questions that arise when the lines between species blur and the very essence of what it means to be human is challenged.